Hello, my dear students. Welcome to my today's online class on one more topic in genetics, that is paper 6 of semester 5, entitled Genetics and Biotechnology of Bangalore City University. And I am Dr. MEERA B K, Associate Professor, Department of Zoology, Maharani Science College for Women, Bangalore 1. And before getting into the details of our talk on gene mutations today, it's my privilege to thank my good friend and colleague, Dr. Lata V, who has prepared these quality PPTs, and my role of making this video lies only in giving the voice. So once again I thank Dr. Lata, and now with this background and with this brief introduction, let us start our class. You can see one photograph here. So, telling you that mutation is nothing but heritable change in DNA. You can see the double helix presentation of DNA, and you can see the cut, right? So, any change in DNA, heritable one, will lead to mutations, okay? And our talk for this class is going to be on gene mutations, spontaneous and induced mutations. Let us see the details now. So, learning objectives for students, you will learn the role of mutations in bringing about variations. Natural mutations, when it happens by nature, right, is the sole source for variations. Okay, it is very much needed also. And one more objective is you will understand the different types of mutations, their causes and effects. So once you are done with your class, you would have learnt about the role of mutations in variations and different types of mutations, spontaneous and induced, their causes and effects. So the lecture includes a brief introduction to mutations, definition of mutation, and a discourse on the types of mutations, namely spontaneous mutation and induced mutations. Now, mutation is an important phenomenon because it is the ultimate source of genetic variation and serves as the raw material for evolution. So, not all mutations need to be induced. Just like that in nature, mutations do happen. If they were not to happen, there wouldn't have been any variations. Okay? So, they happen to be the ultimate source of genetic variation. And that is the fuel for evolution or evolutionary processes. And all the genes would exist only in one form, and you would not find any alleles or mutants. Okay? Without mutation, you will see only one form without any allelic forms. Right? And therefore, mutations are essential to provide new genetic variability and to allow organisms to evolve and adapt to the environmental changes. Then what is mutation? Mutation can be defined as a sudden and heritable change in the nucleotide sequence of a gene. So by now you must be knowing gene is a piece of DNA, right, which is capable of producing a functional protein, and it is made up of nucleotide sequences, right? So if there is a change in the nucleotide sequence, that becomes a heritable change leading to the mutation. By now you know that there are 4 nucleotide bases which go into the formation of DNA: A, T, G and C, right? Then mutations always change the genotype and therefore the phenotype of an organism. In the first chapter you have learnt about the concepts of genotype, phenotype and phenocopy, right? So any change in genetic material is manifested on the phenotype of an organism, right? And that's how you will see the phenotypic changes, right, due to mutations. Then a cell exhibiting change in its phenotype due to the effect of a mutation is called a mutant variety or mutant strain. And not all the time mutations are natural. Sometimes it can be induced by artificial agents. Any physical or chemical agent which increases the rate of mutation is called mutagen or mutagenic agent. Gen mean to produce, muta mean to mutation. So any agent that brings about the rise in mutation rate is referred to as a mutagen or mutagenic agent. Then based on the occurrence, you can classify or divide the mutations into 2 types: spontaneous mutations and induced mutations. Alright, let's see. Spontaneous mutations are those that arise occasionally in the absence of external agents. So mutagens have no role to play in spontaneous mutations. And these mutations may result due to, number 1, errors in DNA replication. Number 2, from the action of transposons or the jumping genes popularly called, or due to the effect of some mutagenic agents present in the environment. Okay. Now, one of the most commonly operating spontaneous mutation mechanism, is transition and transversion mutations. Okay, transition and transversion mutations. In that, first we shall learn about frame shift mutations. So, transition and transversion mutations. The bases of nucleotides in DNA strands exist in tautomeric form. Okay, they exist in tautomeric form. What is this transition mutation? Transition mutation alters hydrogen bonding characteristics of the base following purine to purine or pyrimidine to pyrimidine substitutions, resulting in stable alteration of the nucleotide sequence. Okay. So, if there is change between A and G, right? It is transition, and TA like that, it is transversion. Okay. Now, so transition mutations alter hydrogen bonding characteristics of the bases allowing purines to purine or pyrimidine to pyrimidine substitutions, resulting in the alteration of the nucleotide sequence. On the other hand, transition mutation, here pyrimidine to purine or purine to pyrimidine, case B here in the diagram. Okay, and transversion mutations are quite rare. Then, so here you can see the representation. What exactly happens at the molecular level, the transition and in case of transition and transversion mutations. Okay. Now, so transition and transversion mutations, what happens here is errors in replication of DNA creep in due to base tautomerization. Okay. You can see in the diagram A, normally AT and GC base pairs are formed. Okay. When keto groups participate in hydrogen bonding, but contrary to it, enol tautomers result in AC and GT base pairs. Okay, they result in AC and GT base pairs. This is what happens in transition. Okay, in B, mutation as a consequence of tautomerization during replication of DNA, the temporary enolization of guanine results in the formation of AT base pair in the mutant and a GC to AT transition mutation takes place, right? So, you can see what happens exactly at the molecular level. Then, next type is a frame shift mutation. This is usually caused by deletion of DNA segments. Deletion of DNA segments resulting in an altered codon reading frame. They originate from lesions in DNA as well as from replication errors. For example, it is possible for purine nucleotides to be depurinated or to lose their base. This results in the formation of an apuric site which will not base pair normally and may cause a transition type mutation after the next round of replication. Or cytosine may be deaminated to uracil, which is then removed to form an apyramidic site. Okay. So here is the representation at the molecular level telling you about frame shift mutations, additions and deletions in the complementary strand, that is new strand of DNA. Here you can see a hypothetical mechanism for the occurrence of additions and deletions during DNA replication. The large arrow indicates the direction of replication. In each case, there is a strand slippage leading to the formation of a small loop stabilized by the hydrogen bonding in the repetitive sequence. The AT stretch in this example A. If the complementary strand, that is new strand, slips, an addition of T results and B tells you the slippage of the parental strand that results in a deletion, the loss of two T's, which is an example in this case. So, this is what happens exactly in frame shift mutations. Then the next category is induced mutation. Mutations are induced by any agent which directly damage DNA, changes its chemistry, or interferes with repair mechanisms. Okay, and such mutations are termed induced mutations. Okay, the agents which cause such actions are known as mutagens, and they can be conveniently classified according to their mechanism of action. In my next lecture class, I am going to deal in detail about these mutagens. Okay, right now whatever is necessary to understand the concept of mutations, let me explain. So what are the modes of mutagen actions? So base analogs, these are similar to normal nitrogenous bases that can be incorporated into the growing polynucleotide chain during replication. Analogs are introduced. Okay. Then once in place, these compounds typically exhibit base pairing properties different from the bases they replace and eventually cause a stable mutation. Say 5-bromouracil, 5-BU, is a widely used base analog of thymine. So it undergoes a tautomeric shift from the normal keto form to an enol, bringing about enolization, okay, which happens more frequently. The enol forms hydrogen bonds like cytosine and directs the incorporation of guanine rather than adenine. Okay, instead of adenine, guanine goes and sits. So the mechanism of action of other base locks is similar to that of 5-BU. Okay, then, so this is the representation of base analogs, right? So diagram A tells you the keto form of 5-BU pairing with adenine, whereas diagram B tells you 5-BU is a tautomerized to enol form and pairs with guanine rather than adenine, bringing about the mutation. Alright? Then, specific mispairing. This action is caused when a mutation changes a base structure and therefore alters its base pairing characteristics. Certain mutagens in this category are selective. They preferentially react with some bases and produce a specific kind of DNA damage. Now, methyl nitrosoguanidine, an alkylating agent that adds methyl groups to guanine, causing it to mispair with thymine. A subsequent round of replication could then result in a GCAT transition. Then ethyl methane sulfonate EMS and hydroxylamine are the other examples of mutagens with similar mode of action. Then you can see here specific mispairing. Okay, so mutagenesis by base analog 5-bromouracil. So in the diagram A, okay, the enol form of 5-BU pairs with guanine rather than with the adenine, as might be expected for a thymine analog. Right? This is how specific mispairing takes place. Then next is intercalating agents. These agents distort DNA to induce single nucleotide pair insertions and deletions. These mutagens are planar and insert themselves or intercalate between the stacked bases of helix. This results in a mutation, possibly through the formation of a loop in DNA. Acridins, proflavin or acridin orange, that is the dye, are important intercalating agents. So here you can see the how exactly intercalating agents bring about mutation. This is a plate briefing you about mechanism of intercalation of an acridin molecule in the replication fork. Alright. Then other mutagens which directly damage DNA bases severely are UV radiations, okay, that generate cyclobutane type dimers, thymine dimers between adjacent pyrimidines. Then aflatoxin B1 and other benzopyrene derivatives cause damage to DNA, which is generally lethal but may trigger a repair mechanism that restores much of the damaged genetic material. So, dear students, these are the references what Dr. Lata has made to prepare these PPTs, textbooks and also web references. And then it is the time for me to discuss few assessment questions. So let me discuss. The mutations are mainly responsible for increasing the population rate, maintaining genetic continuity, constancy in organisms, variations in organisms, and answer is D. Next assessment question. Name the type of mutation in which the cause of mutation is not known. A. Spontaneous mutation, B. Suppressor mutation, C. Nonsense mutations, D. Missense mutation. Answer is A, that is spontaneous mutation. Then question number 3 or assessment 3. Addition or deletion of bases causes which kind of mutation? A. Transversion, B. Frame shift mutation, C. Transition, D. Transcription. Answer is B, that is frame shift mutation. Then fourth question, multiple choice question, which of the following chemical mutagen affects only replicating DNA? A. Acridine dye, B. Alkylating agent, C. Deaminating agent, D. Base analog. Answer is B, that is alkylating agent. Fifth question, in mutational event, which adenine is replaced by guanine? A. Transition, B. Transversion, C. Transcription, and D. Frame shift mutation. And the answer is A, that is transition. My dear students, till now I have taught you about the mutation, definition, significance in evolution, types of mutations, reasons for mutations, and a brief account of mutagens, followed by few assessment questions. Thank you for your patient learning. Let me end my lecture today. Thanks a lot. Stay blessed.